In a previous lesson, we learned a little bit about the cerebral cortex and why it is that these regions seem to be doing different jobs. It had to do with their input-output relations. This is visual cortex because it's receiving inputs from the eyes, for example. So the wiring of the brain is functional. The brain cells are doing certain jobs, and different parts of the brain are doing different jobs because of the nature of the inputs, the information they're getting. We also learned there were three general approaches to mapping the functions of the cerebral cortex. We can examine the symptoms of brain damage, we can record the electrical activity, or just when is that part of the brain active, under what circumstances, and then we can electrically stimulate that part of the brain to see what happens. In this lesson, we'll look at how these three techniques have been used to characterize a part of the brain that seems to process faces. In modern times, scientists have identified several regions of the cortex that seem specialized to process certain kinds of stimuli. For example, in blue here, we're on the underside of the brain now, so the front of the brain is here, the back of the brain is here, so this would be the right hemisphere, the underside of the right hemisphere, and the bottom of the left hemisphere here. On the right side, it's in the left too, but on the right side there's a larger region that scientists call the fusiform area, the fusiform gyrus, and they call it the face area because this bit of brain tissue seems to electrically respond when a subject sees face. So they call it the fusiform face area. Other regions of the cortex seem to selectively respond to other kinds of stimuli, the body, or here in pink, the place area. So this bit of tissue here seems to respond to stimuli like houses or nature scenes, etc., places. Whereas in blue, this bit of brain cortex seems to respond selectively to faces. Now, the first evidence that there was such a place, a face area on the cortex, came from brain lesions. So some patients had damage to small regions of that fusiform gyrus, and that seemed to be related to a condition that they suffered called prosopagnosia. They had difficulty recognizing faces, even the faces of their family members. So the suggestion then was that damage to that small bit of a brain area caused these symptoms. Well, if so, perhaps that part of the brain was somehow involved in processing faces. In more modern times, a very widely used technique to study brain function is called the FMRI, or Functional Magnetic Resonance Imaging machine. It's a big magnet. You put your head in there, and the device can measure the functional activity of the living brain. What fMRI is really doing is measuring something that is kind of a substitute for direct brain activity. Here's how it works. All brain cells, here's a neuron here, all brain cells are near to a blood vessel. They must be near a blood vessel because the blood vessel is carrying oxygen and sugar, glucose. Glucose is the sugar that is the principal fuel, energy molecule for cells in the body. All brain cells must be near a blood supply. In fact, brain cells are very energetically active, so they need sufficient blood and oxygen and glucose to keep functioning. Generating those electrical signals and releasing chemical messages, that takes a lot of energy. So brains are very metabolically active tissues. Now, if you just remove a brain from a skull, you'll see it is kind of very bloody, and you can see lots of blood vessels that are bringing blood to every part of the brain. So brain cells absorb glucose and oxygen from the blood. Glucose is the main energy, oxygen needed for metabolism, and here's the key principle. Active brain cells require more nutrients. So, when brain regions that are excited by some stimulus, when they are highly active, it causes the local blood vessels to increase blood flow to that region. So let's say we've got a couple of brain cells here that are, just kind of hanging out, and they're generating electrical signals every once in a while. But then a new stimulus comes along, and these cells are generating lots of electrical signals. Well, when they generate a lot of electrical signals, what happens is locally the blood vessels nearby start to get larger so as to bring more blood carrying oxygen and sugar to service the needs of the higher activity level of those neurons. So the basic idea is when a part of the brain is being activated, the local blood vessels get larger. And this is what the machine is doing. It's going to be detecting changes in blood flow in different regions of the brain as a substitute for, or as a measure of changes in brain activity in a certain region of the brain. We're not going to get into the details, but the idea is blood with oxygen has different magnetic properties than blood without oxygen. So brain areas receiving increased blood flow can be measured. So the functional MRI measures blood flow changes as a substitute for brain cell activity. Nevertheless, it's a very useful technique. It's not measuring brain cell activity directly, but it's a pretty good relationship between blood flow and brain activity in a localized region. So when scientists put subjects in the fMRI scanner and they show the subject faces or objects, they find that this little piece of brain tissue, the fusiform gyrus, starts to become active when the subject sees a face, but not when it sees other objects. So you get brain activity in this little bit of tissue that seems to be selective for faces. And so scientists today call it the fusiform face area. Now there's another way to study the fusiform face area, if it really is processing faces, and that is to do stimulation studies. This is a picture of a brain from the work of Penfield. He was a neurosurgeon some decades ago, and he was a pioneer in brain surgery. And when he would have his patients on the table, awake and conscious, before he would do his operation, he would stimulate different regions of the cortex and then ask the patient what happened. What experience did they have? And so here you see these little numbers here. These are all the places that he stimulated. So this was position number 10. He would stimulate there and make a note of what the patient reported. So brain stimulation, you'll recall, is another way to study, to map the cortex. And in fact, as it happens, this individual, again, going to be undergoing surgery for epilepsy, had electrodes implanted in his brain, and he happened to have some implanted in the fusiform face area. So scientists took the opportunity to do a little experiment. They wanted to see what would happen if they stimulated the fusiform face area. And sure enough, something interesting happened. Stimulation of the fusiform face area produced face perception disturbances. So while he was looking at his doctor, the stimulation proceeded, and the patient reported that the doctor's face appeared to sort of morph into some other kind of face. The face changed its identity, as it were. So this is evidence then that stimulating the fusiform face area was altering face perception. So it seems then that scientists have discovered that the human brain has a bit of tissue on the underside of the temporal cortex called the fusiform face area that seems specialized for processing human faces. Of course, all visual stimuli enter through the eyes, and that information reaches the visual cortex. But further along the processing pathway, there is this area of the brain that seems to be getting just the right inputs that makes it specialized for processing human faces. And we've seen the damage to this area impairs facial recognition. If you stimulate this area, it alters face perception. And if you simply record when is this area active, it's active when people are seeing faces. Now, it is technically possible to record the electrical activity of one cell in the brain. This is done in animals all the time. In humans, it's just more rare because generally we don't stick electrodes into humans that don't have any reason to have electrodes stuck into their brains. But sometimes you'll have the epilepsy patient that will agree to having an experiment performed on their brain. But we would predict then that in the fusiform face area, we would find cells that have a preferred stimulus. They become activated when a certain visual stimulus is presented to the subject versus another. So let's say we were recording from a cell in the fusiform face area. What we would predict is that if we show some object on a screen, that cell would not respond. If we show some nature scene on the screen, that cell would not respond. Rather, if we show a face, that cell would respond. So we predict that in the fusiform face area there would be cells that are individually responding maximally to faces as their preferred stimulus. And we call this the preferred stimulus then of a neuron is whatever stimuli or stimulus that generates a maximum response, the maximum number of the highest frequency electrical signals. And you'll recall in an earlier lesson that neurons can carry information by adjusting the frequency of their electrical signals. In the case of sound, if it's a soft sound, low frequency signals coming from the ear, loud sound, high frequency. Dim light, low frequency, bright light, high frequency. Well, in the case of a cell in the fusiform face area, the idea is it will be quiet if there's no face in the environment. But when a face shows up, that cell will respond then. That's what it would mean to be a face-selective cell. It has a preferred stimulus of a face. Other objects just do not excite that brain cell as much as faces. So this is an example of how the activity of a brain cell means something. Activity in this cell would mean there is a face in the environment. And this is how brains traffic in information. It's the electrical signals that are the language of the brain. And when cells are making lots of electrical signals, it means that they have received just the right inputs and their activity means something to the next part of the brain where the output signals will be sent. So this cell's activity can signal other brain areas that a face has been detected. And so modern techniques have helped to characterize the functions of different parts of the cerebral cortex, looking at the results of brain damage, stimulating different parts of the brain, and recording using either FMRI or actually electrodes to record the activity of groups of cells or even individual cells. All of these modern techniques have helped scientists map out the cerebral cortex and the functional specializations of different parts of that cortex.